All right, this is the last of the videos that I'm going to make regarding immunodeficiencies. In this case, we're going to discuss immunodeficiencies that affect the innate immune response. If you have questions regarding the other immunodeficiencies, I've made a video for the B cell compartment, the T cell compartment, the combined compartment, and then also a video that provided an overview of all the primary immunodeficiencies. All of those are available on this channel and you can check them out. So for the immunodeficiencies of the innate immune response, we're really talking about a couple of things. How do the cells get there? And how do they kill? If it interrupts either of those things, then that's an innate immunodeficiency, okay? So the first one we're going to talk about are basically the leukocyte adhesion deficiencies. Neutrophils are the primary phagocytic cells in our host defense arsenal. There are three major functions of the neutrophil, and it needs to perform all of these in order to protect us from invading organisms. One, it has to migrate. Once there, it has to phagocytose. It has to eat. And once it's phagocytosed, it has to kill. It has to undergo oxidative burst, okay? So we're going to start with step one, migrate. In leukocyte adhesion disorder, this normal migration is not intact, okay? So as cells move down the highway that is your bloodstream, they have to go through these steps that basically slow them down before they can extravasate through the epithelium to get to the site, okay? This is basically controlled by adhesion molecules. And in leukocyte adhesion deficiency, CD18, which is an adhesion molecule for the beta-2 integrin, isn't working, okay? So this is an autosomal recessive issue. In LAD, this gene is missing or defective. So therefore, the adhesion molecules are not present, and now the neutrophil can't migrate into the site. So we wind up with severe infections, umphalitis, and basically you're going to diagnose it by high polymorphonuclear cells in the blood. They're being released from the granulocytic reserve. They just can't get to the site, okay? The infections are going to be in all sorts of places, lungs, liver, etc. The infections are severe and life-threatening because basically they can't be combated. LAD usually presents in the first days to weeks of life with pneumonia and sepsis. This is actually a patient with severe life-threatening cellulitis. You can see it's inflamed, but not because of our cells. A striking unique feature of LAD is actually that there is a delay in the separation of the umbilical cord from the baby. The cord should fall off in a couple of days after birth, but in LAD, the cord stays attached for up to 2 weeks, and the umbilicus is severely infected, and this is known as umphalitis, and it's what's shown here. To diagnose this, you're really just looking for those high PMNs. Then you can do flow cytometry and basically look for the absence of CD18. When you don't find that, you pretty much know what's going on. The only curative treatment is a bone marrow transplant or a stem cell transplant. All right, the next thing is, well, what do we do if the cells get there, but they can't kill, okay? Before microbes can be phagocytosed by neutrophils, the microbes must be opsonized. The two most important opsonins are IgG and C3b. After microbes are phagocytosed, they are then killed by either enzymes from the granules of the neutrophils or by reactive oxygen species generated by neutrophils via the NADPH oxidase pathway. Patients with CGD essentially have a mutation in one of the genes that encodes for the subunits of the NADPH oxidase, okay? And if you don't have that, then you can't make NO or any of the other byproducts that you need. It can be X-linked or it can be autosomal recessive because there are several subunits in the NADPH protein. One of the subunits is encoded by the X chromosome and the other are encoded on different autosomes. Patients usually present at an early age, 1 to 5 years old. They have severe skin abscesses, they get cellulitis, pneumonia. You're starting to hopefully paint a picture now of what kids with innate immune deficiencies look like. Liver abscesses or osteomyelitis are pretty common. The infecting organisms tend to be catalase positive organisms. They tend to have the ability to survive really well in this. So specifically, we're really worried about staph infections, right? How are we going to diagnose it? Flow cytometry is great for this one. Basically, we use a molecule called dihydrorhodamine, or DHR. DHR will fluoresce when it's oxidized. Neutrophils are mixed in a solution containing DHR, and then that basically will diffuse inside the neutrophil. And then the neutrophils are stimulated by a mitogen. Normal neutrophils, when stimulated, will produce an oxidative burst, okay? So you can see that if you have an unstimulated patient and then you stimulate, you see a nice jump. If you look at the mother of this patient, nice jump, right? This is high DHR because her cell's oxidized. In the patient, no oxidation occurred. We don't see a high fluorescence of DHR. So it's kind of a very clean system for diagnosing this. Okay, the last one we're going to talk about are the complement deficiencies. There's a whole bunch of them, C1 through C9. Basically, a terminal complement deficiency will make you more susceptible to Neisseria infections. Remember, those are specifically C5b through C8. There's actually no effect of a C9 deficiency. C5b through C8 basically make up the scaffold for the membrane attack complex, or the MAC attack. If that doesn't happen, then we can't kill some of these gram-negative infections, and Neisseria are the most common. So the one we're most worried about is Neisseria meningitidis, but I can't say I'd exactly be excited to experience Neisseria gonorrhea. How do we diagnose it? You use a complement hemolysis 50% test, or a CH50. This basically stands for complete hemolysis. The test is performed by mixing a test tube in a solution of sheep red blood cells that have been attached with rabbit-derived IgG antibodies. So here's my sheep red blood cell. Here's my rabbit-derived IgG. The patient's serum is then added to the test tube, okay? Because the patient's serum is missing one of the components of MAC, there is no way to puncture this red blood cell and make it explode. So therefore, it won't turn the test tube. The test tube will stay clear. A normal person's serum would form the MAC and the red blood cells would undergo hemolysis and the tube would turn red. But in a patient who is lacking any of these components, we won't see that. There's no cure for the disease. The patients need to be treated with IV antibiotics at the first signs of meningitis, but we tend to do that anyway. Also, these patients should definitely be vaccinated against Neisseria meningitidis and encouraged to practice safe sex.